to start at 12:10 as we always do. Okay, why don't we get started? Welcome to MVZ Lunch. It's great to see everyone, virtually at least. Before I introduce our speaker today, are there any announcements? Okay, next week, I'll just remind everyone, well, two things. Carly's been sending out seminar announcements for other seminars, I think on Monday of each week. And so be sure to check those out. Normally we announce those at the beginning of seminar, but we're doing this by email now. Next week in MVZ Lunch, we have Uns Rasafindratsin, who's going to be speaking on lemur seed dispersal in Madagascar's rainforests. Uns is a new professor in IB and is being considered as an affiliated faculty member of the MVZ. So I hope everybody will be able to attend that seminar. And then the next two seminars after that are from new postdocs in the MVZ, Austin Patton and Simon Scarpetta. So the schedule is on the website. Please look at that to see what's coming up. We haven't made decisions about the format for next semester. Certainly for this semester, we're going to continue online like this, but I'm hopeful that we can get back in person soon. Well, it's a real pleasure today to introduce Yocelyn Gutiérrez, who I trust many of you know. Yocelyn is a postdoc in my lab, and she received her PhD from UNAM in Mexico and is a UC Mexis postdoc. She received this award quite a while ago, but was waylaid by the pandemic, like so many people, and stuck in Mexico until she was able to join us this summer, just a short while ago. So we're thrilled to have you here, Yocelyn. She sits in the office right next to me. So if you haven't met her, please come by and introduce yourself. She did her PhD with Luis Eguiarte, and she worked on phyllostomid bats. So she sequenced the genomes of a number of phyllostomid bats, the complete genomes, and leaf-nosed bats as a family are remarkable for their dietary diversification. So they're frugivores and nectarivores and insectivores and blood feeders, vampire bats. So there's this tremendous radiation of ecological forms, and she was interested in exploring the genomic basis of that in her PhD work. So presumably we're going to hear a little bit about that today, as well as some of the work she's doing here on environmental adaptation in mice. So Yocelyn, welcome, and I will turn it over to you to share your screen and tell us all about bats and mice.
Thank you, Michael, for this kind introduction. So yes, well, today I prepared for you a talk with two different tales, independent tales, and I call this talk about genomic change in nectarivorous vertebrates and environmental adaptation in wild mice population in North and South America. So before I start, I would like to introduce briefly myself. Michael mentioned a lot of my background. I just want to say that I'm from Mexico City. I study biology in the Universidad Nacional Autónoma de México, UNAM. I don't know if you have been the opportunity to be there, but it's an amazing campus. And if you go to Mexico City, please go to the south of the city and visit all of these campus because you will enjoy it. So the UNAM is a very important institution in Latin America, and you can find a diversity community in this place. And also, well, I study my biology in the Faculty of Science, but also I did my PhD in the same university, in the Instituto de Ecología, in the Laboratorio de Evolución Molecular Experimental, with the professor Luis Eguerte, who is a passionate of population genetic, evolution, plants, and the ecological relationships between bats and agave. And that's why I started working with bats and another vertebrates. So, well, during these years, I had the opportunity to establish many collaborations in different projects, including projects with another mammals, with the prairie dogs, plants for the teocintle, the wild relative of the corn, the domestication of the squash, and well, and then I start to think about my PhD, and I established a great collaboration with Dr. Martinez del Rio, Carlos Martinez del Rio, who is in Wisconsin working, and we start to think about the idea to how to live and just eating sugar. So we start to think a lot about the guts and the sugar. So it's why I want to show you in the first tale that I prepare for you. So the first tale, it's about the genomic change in nectarivorous vertebrates. But before to go to this sugar talk, I briefly want to share with you a part of the background that how can we generate all of this data that I'm going to present. So during my PhD, I was interested in investigate the genomic and evolutionary dynamics associated with the dietary diversification and nectar pollen feeding specialization. So Michael mentioned that this, and I'm going to mention in a moment that this bats of family, it's huge and amazing because we can trace in this family different change in the diet. So now I'm going to show you three predictions that we explore in this first tale. So the first part is the dietary diversification um, from in the phylostomid bats from the insectivory, who is the ancestral condition of all of this group, to the derivatives diets that will be accompanied by evolutionary change in relevant genes involving in the food uptake and also metabolic pathways. I'm going to talk to you in a moment about it because also include many pseudogenization and acceleration in substitution rate in some genes. Then I want to move the talk in a specialized group, the nectar, pollen, and fruit feeding bats. And I'm going to talk to you about the adaptative signals and parallel evolution between the North World nectar and fruit feeder bats and the Old World fruit and nectar bats. And finally, we are going to I'm going to show you the nectarivory evolution in two hovering vertebrates, in particularly in hummingbirds and nectar pollen feeder bats, using RNA-seq data and also genomic data. So, well, the leaf nose bats are a good model, as Michael mentioned, to study the dietary diversification and specialization because they evolved from an insect feeder common ancestor and now displays a large and diverse spectrum of feeding habits that include the insectivory, the carnivory, the frugivory, and omnivory. And two subfamilies has a extraordinary specialization. I think my favorite specialization is the vampire, but also we have another group of bats from the subfamily Glossophaginae who are able to survive only drinking nectar and a small amount of pollen and some insects. So many studies have demonstrated evidence of evolutionary novelties associated with this feeding diversification, but also with this specialization that include morphological, physiological traits, and some genetic change. So in order to explore and to trace the genetic change in this diversification and radiation of the different diets, during my PhD, we sequenced the whole genome of 5 phylostomid bat species with different diets. We sequenced the whole genome of the fruit bat, Artibeus hamisensis, the 3 nectar pollen feeder bats, Musonectaris harrisoni, who is this bat who has a very, very long tongue. I like to say that it's like a hummingbird, but from the Chiroptera order. And also we sequence the whole genome of Leptonicteris yerbabuena. For this species, we have a special attention because it's the main pollinator of a different agave plants. Also sequence the whole genome of the sister species, Leptonicteris nivalis, and the genome of the insect feeder, Macrotus waterhousei. So we assembled and annotated all of these genome and also incorporate the genome available in database of the vampire. And as you can see, we have a very nice spectrum of the feeding habits. So we use all of these as a genome assembly, the novel genome assembly, to perform a different analysis. So first, we uh, we perform a gene expansion family analysis using all the genome of these bats, the phylostomid bat, but also we include the genome of another bat species from different families available in database, and also more than 30 mammals species, just to have different outgroups. So first, we start with the gene expansion and contraction analysis. Then we look for single copy genes, orthologous single copy genes, along all the genome to perform selection tests, the rate, evaluate the rate of DNDS. And also for specific genes, we also calculate the molecular substitution rates. And for those genes that show it signals of positive selection in our group of nectar and fruit feeder bats, we also generate an extra analysis to reconstruct the ancestral sequence and look for parallel substitutions in amino acids along these specific genes. I will show you in a moment. So to understand the fish bat, and to trace the chains associated with the dietary diversification and specializations, well, first analyze the gene family expansion and contraction along the phylogeny. So this is all for this analysis is all the species that we use. Here you can see our phylostomid bat that we generate, and also we include another bats available in database and some outgroups, mammal outgroups. So the first that we found is that for all the phylostomid bats, we found significant gene family enrichment functions related to the cellular repair process and the genetic makeup for protein synthesis, but also many families associated with immune system. That these results later were corroborated with another project, including more than 30 genomes of only bats. So it was very interesting to see all the immune system machine that have all of these species. And now it's more interesting in these COVID times. But regarding to the diet, we found that many gene families exhibit change with feeding habits. For example, the insect feeder bat, Macrotus waterhousei, show a contraction in the lipid metabolism. So this is a main insect feeder bat. And moving to the specialist bats, we found that the vampire had a significant gain of gene family involved in the regulation of appetite and the process for nitrogen acquisition, but also to remove the salt. And this is very interesting because the blood contains a lot of salt, so they have this mechanism to remove it. And it's for this species, we also found many pseudogenizations in their sensory receptors, the olfactory and taste receptors. And it was very interesting because another studies have been showing that some pseudogenization in the sweet receptor, sweet taste receptor, and also we found a pseudogenization in this receptor, but also in the umami receptor. So the vampire has changed a lot their system sensory. Uh, we hypothesized that it was just to this change in the diet. And for the nectar and fruit bats, we also found a very interesting expansion in some gene families involving in the iron metabolism. And it's very interesting because the nectar is a sugar solution that it's poor in nutrients. So these bats, for example, the nectar pollen may take um, pollen who has the essential amino acid, or also they could ingest some insects, but the proportion is very low. So they need to have these resources to keep the iron and don't have any metabolism problem, as anemia, for example. And moreover, we perform. Oh, sorry. We perform an analysis looking for orthologous single copy genes to test positive selection in different coding sequence genes. So we perform this analysis for each of the species and in more than 8,000 orthologous single copy genes. And for each bat with a different diet, we found specific things, specific genes involved in metabolism under positive selection. But I want to stop here just to show you that for the vampire, we found interesting genes associated with avoid the blood coagulation. So this is very important for if you are drinking only blood. And also, as I mentioned before, that we found that this expansion in a gene family to eliminate the excess of salt, we found some of the genes under positive selection. And moving to our next prediction about the nectarivorous evolution, we found for this group the Glossophagidae and nectar pollen feeder bats, Musonectaris harrisoni, Leptonectaris nivalis, and Leptonectaris yerbabuena, a group of positive selection genes that are part of very important metabolic pathways as the glycolysis or glucogenesis. And here in this diagram, you can see in red all these genes and enzymes that are under positive selection and plays a very important role in these pathways. So it's very interesting because just 2 weeks ago was published another a paper exploring the same question, and they found the same enzymes and the same proteins. So it's very great that well, these results are consistent. Well, now we found again some genes involved with the efficient year room storage, from involved with the carbohydrate metabolism and also with the lipid and fatty metabolism, only for the New World and phylostomic bats. So then we was thinking about a convergent evolution into the Chiroptera order between the New World nectar pollen feeder bats and the Old World fruit bats. So we perform an analysis just to again looking for orthologous genes and perform again our selection test in more than 5,000 genes, and we found these three genes that are under a strong, that show a strong positive selection and are very important in the ketogenetic metabolisms, and also in the lipid metabolisms, because these genes are important in the transition to convert all the lipids or all the fat to energy. So this is what for us this was an extraordinary result, because uh, it's a notable, because given that the theropodid and the glossophaginal lineage are separated by over 60 million years ago, this convergent evolution may be a consequence of the dietary specialization and the high metabolic rates that I need to fly every day, and this constriction in their diet and feeding resources. And also, we identify an olfactory receptor on the positive selection that is sharing for the phylostomid fruit and old world bats. So, uh, and and for this receptor, we found positive selection in these fruit bats, but in the vampire, there an acceleration and increase in their substitution rate. So, probably this olfactory receptor may be linked to the perception of some components from the fruits. And the next part of this tale, is the last part of this tale, is in order to explore the challenge posed by a nectar diet, rise, rise the question about of whether different lineage of nectarivorous have converged in morphological, physiological, and biochemical traits. We include in our analysis the birds, because in general, all beers are similarly hyperglycemic, and they have a higher blood sugar levels that appears to be independent of the diet. But following their good, uh, we can we in we can explore the three major nectar feeding birds clades. We have, for example, into the birds, the hummingbirds, and also the songbirds and the honey eaters. But there are species like the flower pieces who has a diet dominated by sugars. Um, but into the birds, the human birds show extremely metabolic uh, extreme metabolic rates, even, and they have the ability to metabolize sugars, a lot of quantity of sugars, than the other birds. And comparing with the bats, human birds and bats became nectarivorous animals in a process that involved the coevolution with the other flowering plants. And also, this coevolution between hummingbirds and nectar bats involve ecological, this coevolution with the specific plants, but also physiological convergence in the different sugar preference, but also in biochemical pathways. And uh, this part of the project was very interesting because
She seems to have frozen. She's in the office next door. Let me go grab her and let her know. Let me see if. Okay. Okay, why don't you get out and get back in? Everything is perfect. Okay. Stand by. She logged out and will log back in to Zoom. Everything was good until then, but this happens occasionally. Yeah. Rory is saying air bears is still having many issues. Isaac's saying some of my students in a lecture this morning were also reporting campus Wi-Fi problems. There have been a number of emails about this. If you're on campus and can connect with a hard line as opposed to Wi-Fi, that always seems more secure.
So Michael, obvious question, are you on a hard line connection right now?
Yep.
Yes, yeah. You're
Yeah,
so good.
I essentially never use Wi-Fi when I'm on campus. I think in the MBZ there are hard. Oh, there she is.
Yeah, sorry.
All right, you're back. We can hear you.
Yeah, sorry. I'm going to share again my. Oh, could you help me to be a host?
Yeah, you should be able to now.
Yes. So, well, there are different evidence, ecological, physiological, and biochemical, that show that hummingbirds and nectar bats has a lot of convergence, but have not been exploring with a genetic data. So in this part, I want to show you these results that we are generating in collaboration with a group of Mexican students and professor, and also a professor from Wisconsin, about how do hummingbirds and nectarivorous bats avoid the negative effects of the highest blood sugar glucose levels, and how can they live all their life or most of the time of their life feeding only nectar. So in this part, we generate the whole genome sequence of different hummingbird species, the genome for the Iliocaris leucotis, from Colibri thalassinos, but also for the flower pizzers, the Glossa varitula. Also for Iliocaris leucotis, we generate the genome and we sequence the RNA-seq. And we first analyzed the parallel gene expression in fatty and sugar metabolisms, comparing the transcriptome genome of Iliocaris leucotis and our previous results with the bats, with the species Leptonicteris yerbabuena, to perform analysis just between these two species, both species. But also then we incorporate the RNA-seq of the of liver data available in database for another 8 hummingbird species and another insect feeder bats and birds. Evaluate the differential gene expression and perform this comparison between to identify parallel gene expression between nectarivorous birds and bats. And then we use the genome, the whole genome, to look for molecular convergence in specific metabolic pathways. We perform a gene family analysis and also a selection test. So with the RNA-seq, we analyze all of these liver transcriptomes, and we identify that we assemble all of these liver transcriptome and perform the gene differential gene expression between two groups. We have the insectivorous and the nectar and fruit vertebrates, and we found in our analysis that there are many genes that show it a high that I differential expressed between the pollen nectar vertebrates in contrast with the insect pollen feeder. So it was pretty interesting identify that there are different genes that we found previously only in the bats on the positive selection and with these signals of duplication in their gene family evolution showing this differential expression. So we found again a group of genes involved in this conversion and this metabolic pathway that convert lipid to energy. And then analyzing the whole genome sequence, we found that the gene family reflects the distinct dietary needs. So in general, for all the birds that we included and analyzed here in this gene family analysis, most of them are involved in immune system, make our proteins, cellular repair, the same for the bats, but paying attention only to the hummingbirds, we found expansion in some families that are linked to skeletal muscle and the myosin that is more for a morphological and skeleton movement. The iron storage, again, we found it for the hummingbird as we found previously with the bats. Different calcium transporters and a lot of enzymes involved in the glucolysis glucogenesis pathway. But when we include the other nectarivorous bird, the Diclosa varitula, we identify for both groups some enzymes in the family of the aldoketo reductase that it's important to convert again lipid and sugar to energy, and also the ammonium transporter. But we was for us was very interesting identify in our positive selection test using all of these genome uh, that this protein, the GLUT12, is on the positive selection for the nectar pollen vertebrates and also for the fruit bats. This is very interesting because the glut there is a proposal about that how these hovering vertebrates can convert very quick all the energy that they use that they are feeding in energy, and that's why they can metabolize and oxidize the sugar that they uptake in energy as around the 100% of efficiency. So this protein, it was very interesting because it's important and is the second sensitive insulin transporter, and and this is expressed in skeletal muscle, and these proteins send the signal to avoid the homeostasis of sugar in blood. So a lot of people who has a problems with diabetes, for example, they have a mutation or problems in the GLUT4 and the GLUT12. So found positive selection in this protein only in these nectar pollen feeder vertebrates. It has been really interesting for us, and we are now analyzing in detail and using metabolomics and biochemical analysis. Uh, uh, these analyzing these proteins in some hummingbird species. So, if well, I'm still learning. For we found here that there are convergence that at different levels, ecological, physiological, biochemical, but also genetic convergence. So the specialization in a diet dominated by high sugar concentrations have evolved mechanisms to avoid the glucose toxicity. And these mechanisms has evolved by gene family duplications. Also, some of these genes that are duplicated showed high expression with the RNA-seq data, and also parallel positive selection in some of these proteins. So I'm still largely ignorant of the evolutionary causes of the nectar feeding condition, but and also its repercussions for the in a metabolic physiologically point of view. But I think that it's really important to explore the gene regulations mechanisms and the dosage of these genes in the future, just to have more information about this real convergence and also how can live on sugar most of their life. And I want to move to my second talk because during I think this postdoctoral stay and all of your experience at the Nachman lab and all the museum, the people from the museum will help me to learn this approach to explore more about the gene regulation that I can use later to back to the sugar. So in this second tale, I just want to mention some of the work that I have been doing during the last month, and that have been working in collaboration with Megan Pfeiffer, and it's about the environmental adaptation in wild mice population in North and South America. So the rapid spread of Mus musculus of the Mus musculus domesticus in association with the human movements provides a powerful opportunity to study the genetics adaptation in the wild. So Mus musculus domesticus, the house mice, is a successful species who can live in all the world, a cosmopolitan species. And in the Nachman lab, there are having generated and found strong evidence that house mice population have the potential to be locally adapted to different environmental conditions. Even that they have invaded relatively recently the Americas and the North Americas. So uh, recently, there are new paper published this year by Caitlin Ferris, and also the Megan research about the genomic basis of environmental adaptation in house mice showed that in two latitudinal transects along North America in the east and the west coast, in both analyses they found candidate genes involved with environmental adaptation, but linked to traits among populations. So these strong candidate genes, some of them are unique, but also share response related to body size and different metabolism pathway. And I think that these results are very interesting in analyze how this species responds to different selection pressures, but what are those unique and shared responses? So we have a lot of information for North America, but now one of the work that I have been studying, is identify candidate variants that contribute to environmental adaptation in house mice population along the latitudinal transect in South America, exploring some populations from Brazil to Argentina, and also compare all of these candidate genes to identify unique and shared responses to environmental adaptation in America, including the North and Central and South America samples. So I just want to show you some preliminary results that have been generating these last months about the analysis of along this latitudinal transect, um, and the test of selection that I have performed using LFMM2, these latent factor mixed models that implements a a Bayesian algorithm. So for the population from South America, we found that there are more than 500 of candidates. But it's very interesting that most of these candidates, when we filter by some parameters, are involved with different metabolic pathways. But we found that 74 of all of these candidates have been identified by Megan and have been reported by Pfeiffer Rixey in the East Coast, in this East Coast transect. So one of the there are sorry. Oh, there are 74 of these genes that overlap with the previously reported, but it's very interesting that two of them that also showed and Megan reported that as a candidate genes with different expression and allele specific expression are also under positive selection in the South transect. So this one of these genes is MC3R that are highly correlated with latitude. And this is very interesting because in an independent colonization and independent analysis, we are finding the same signals in specific genes. Then we are going to move to the next part of the analysis because we have been paying attention in the exploring just the South America transect, but what's grown in the along all the Americas that it's our next goal to explore. If there are common and unique, if there are unique candidate genes, or we have a lot of shared candidates genes under positive selection. So for this analysis, we are using 185 individuals, including the West and the East Coast, and some populations from Brazil and Argentina. So as you can see, there are some admixture in some population, but we have also a specific genetic pools for each country. So our next steps is compare the candidate genes to identify these unique and shared responses in these gradients, performing and analyzing different bioclimatic variables as the annual mean temperature, the precipitation seasonality, the seasonality temperature, to identify these genes that are unique and can help us to understand the successful colonization of the mus musculus in all America. So this is, I'm still working in this data, and it's part of my goals of my PhD, of my postdoctoral stay here, but also I'm going to start to analyze the model gene. I'm going to start and build a model to generate an analysis for genome-wide association to morphological traits, analyzing different and all the exon capture data available and generated here in the Nachman lab in the museum, and including another data from musculus reported in database, just to generate and analyze this association between body size and the genetic basis, the evolution of the genetic basis. And finally, as a part of this postdoctoral work, I want to explore the expression variation, identify genes that their expression is changing in a clinal fashion with latitude using the SNP, but also the cis-QTL, just to have a complete perspective about the genetic plasticity behind the all of this adaptation and the successfully colonization in house in musculus domesticus. And I want just to thank you for your attention, and sorry for the technical disconnection with Zoom. And also I want to thank all the all my colleagues, all the people that I'm knowing here that I will meet, and I'd be happy to collaborate with you in the future. Thank you.
Wonderful, Yocelyn. Thank you very much. Please join me in clapping virtually. And I'm sure that Yocelyn would be happy to take any questions, either by
Yes.
unmuting yourself or in the chat. Well, yeah, Yocelyn, I have one question, which is, in your analysis of the genome sequences of bats, you set up the study mostly to look for genomic changes that were associated with dietary specialization. But as so often happens in these kinds of studies, you found a strong signal of selection on immunity genes.
Yeah.
And this is a common observation in molecular evolutionary studies. The genes that often show up as showing signatures of selection are those involved in reproduction and those involved in immunity. Of course, right now there's a tremendous interest in immunity in bats as reservoirs for all sorts of things. And one of the things that strikes me about the phylostomids is, in addition to dietary specialization, there's a lot of variation in colony size. Some nectar feeding bats are famous for having maternity roosts with thousands of individuals, and others have fairly smaller colony size, tens of individuals. Have you thought at all about asking whether there's signatures of selection on immunity genes that correlate with colony size, and not only in phyllostomid bats, but across bats more generally, because there are, other species and other families that have enormous colonies and others that have very small group living. One might predict that those in large colonies transmit pathogens more easily and so show strong signatures of selection on immunity genes. You're sitting on a bunch of data that might be used for this sort of thing. Have you thought about that at all?
I haven't think about about colonies. I'm not sure. I haven't thought about it, about the specific colonies, but you are right that in the same cave, there are millions of bats from the same species or from different species living in different cameras. But about the immunity, I had a collaborator, Diana Moreno Santillan. He is working with immune system in Stony Brook. And we analyzed we perform an analysis in all the bats, in more than 30 species of bats, using their whole genomes. This analysis, it was looking for this immunity, the exceptionality immunity in bats. And we found expansion and positive selection signatures in genes involved in the epithelial defense. So probably in these kind of colonies, these genes may be important because it's like the first contact. I think so. But it's very interesting to consider how is, and I think we need more population data to analyze this evolution in immune system in the colonies.
Yeah.
I'll ask a question. It was a very nice talk. I know this is probably not your focus of your study, but I noticed in one of your slides, as an endocrinologist, I noticed in your slide you had a difference between birds and mammals. Was you had insulin listed under mammals and glucagon under birds, and I know in humans there's a complex interaction between insulin and glucagon. So can you elaborate more on the difference between the use of insulin and the use of glucagon for regulating glucose?
Glucose. Yes, well, it's very interesting because in the bears, apparently the bears, he cannot regulate insulin because they lost a lot of these. For example, the GLUT4, this transportant that is sensitive to insulin, is lost in birds. So that's why it was the first question about why birds exhibit high levels of blood of glucose in blood if they don't have this protein that it's very important in mammals. In mammals, is the main protein who sends the insulin. So for us was very interesting to identify the GLUT12 that is under positive selection and appears to be also sensitive to insulin in birds, even when they don't have the main GLUT4 that is for all the mammals is the most important. So probably this protein has a very interesting role in how these birds, in general, all the birds, not only hummingbirds, manage these high levels of sugar on blood. But specifically for hummingbirds, well, we found signatures of positive selection, though, so it's very interesting. And another thing is that the hummingbirds, this hovering flight is very expensive in terms of energy. So hummingbirds and also some nectar pollen feeder bats who has this hovering, they can use all the sugar that they are intaking. They very quick can convert this sugar in energy. And the efficiency, they use around the 80% of all the sugar that they intake to convert in energy. So this is maybe another physiological and biochemical strategy to don't get diabetes or some metabolic transform. Yeah.
I never heard that. So they're kind of permanently diabetic.
Yeah.
Birds.
Yeah. Well, their glucose levels is like for humans. It's like to be a diabetic person. Yeah.
Thank you.
Yeah. Thanks for your question.
Chris, did you have a question?
Sure. Yeah, such a cool talk as always, Yocelyn. Really neat systems and neat work. And I think maybe I missed this. I mean, 100% of the diet of vampire bats is blood, right? Is that? And then I guess I'm kind of wondering, I mean, how did the ancestor was probably an insectivore? I mean, do you see any clues in terms of positive selection or gene family expansions? Like, how did this start? I think we're thinking along similar themes here, right?
Yes, it's very interesting because also some of the genes that I found in the vampire, I found in the finch bird, the vampire bird also, and it's most of their diet is 100. It's not completely one blood because they also take some insects when they bite to the cows or another mammal, but we found a lot of pseudogenization in sensory receptors, but also in many genes. I think it's the bat species with more pseudogenes, and it was hard for us to analyze the data because of the time it was a pseudogene. So probably, I think will be necessary has a genome with a better resolution and another kind of analysis, more like physiological analysis, like experimental analysis, just to trace how they can I don't know the difference between an insect feeder and also the vampire. But it's very interesting, the vampire. I think it's one of my favorite species because they show adaptations at metabolic level. For example, these genes to remove the salt, but also to the sensory system levels.
Okay. Well, thank you again for a wonderful talk. I noticed you had a picture of convergence of Mexican Coke and
Yeah.
US Coke. I'm not sure if that's convergence or
No,
retention
it's a
of
difference of
ancestral. Yeah.
preference sugars.
I don't know about the rest of you. I think Mexican Coke is actually much better. All right. Well, if people have other questions for Yocelyn, you know where to find her. She's in the MBZ in the office next to mine. And thank you once more for a wonderful talk. We'll see you all next week.
Thank you very much for your attention.